Hi, I'm Bob Ward. I'm President and CEO of DSM PTG. That's part of DSM Biomedical. Um, DSM PTG is a vertically integrated developer of materials-intensive medical devices. We have a capability that allows us to design new materials and bring those materials through the scale-up stage to Class 3 medical devices for implantation. One of the areas that we're really excited about is a joint development project we're working on with a local company in Berkeley, California, actually occupying the same campus site that we're on 7th Street in Berkeley, to develop an affinity therapy device that has potential in a lot of infectious and inflammatory diseases. This technology works with a cartridge that's filled with polymer beads, and the surface of those beads is treated with heparin. Heparin is well known for its use over the last 50 years as a very safe anticoagulant to prevent clotting and treat clotting disorders. It has a very short half-life in the body, so it's useful in the clinic. But more recently, it's been discovered that heparin has the ability to bind many pathogens, viruses, parasites, and bacteria. In addition to binding these pathogens, heparin can bind cytokines. The body produces cytokines during inflammatory reactions in the response to disease states. Sepsis, which is a leading cause of death in this country and worldwide, occurs when the body actually produces a flood of these cytokines in response to a serious infection. The cytokines can then damage the organs and lead to multiple organ failure, which ultimately becomes the cause of death. So far, pharmaceutical approaches to curing sepsis have been largely unsuccessful, and many clinical trials have been done using drugs to treat this condition. But because of the unique properties of heparin, it's been used immobilized on polymer surfaces to improve the safety of extracorporeal circuits like cardiopulmonary bypass, oxygenation, and dialysis. So the surface of our adsorption beads are inherently very safe in contact with the blood. But the ability of heparin to bind other components of the blood that contribute to the disease or are a response by the body to the disease is what we use in this affinity therapy. So basically the way it works is this cartridge, about the size of a dialysis cartridge, is put into an extracorporeal circuit outside the body. The blood is delivered to the cartridge and then returned to the body, minus the pathogen and minus much of the cytokine concentration that entered the cartridge. And because of the inherent safety, it's a very simple thing to implement relative to other extracorporeal circuits that are not heparinized. So we're at the point now where we've done a lot of in vitro work, and we're preparing at DSM PTG a large run of cartridges for preclinical and clinical testing, which will occur in the first half of 2011, if not before. So the potential of this device is it may be a safe and effective treatment for sepsis where there doesn't exist one today. And because it can bind so many different pathogens, as I mentioned, parasites, bacteria, and viruses, it could be used to treat a number of other diseases as well. And another exciting application of this is potentially in increasing the safety of the blood supply. During the collection of blood, a miniature version of this cartridge could be positioned between the patient and the blood bag into which the blood is being collected to quote, purify that unit of blood. And even though the blood may go on to be fractionated, it will have gone through this purification step. So in terms of the potential for surface-engineered biomaterials to be used in therapeutic applications, this is a very good paradigm for a number of different future uses of biomaterials. Whereas biomaterials in the past have been used for augmenting or replacing or repairing body parts that are damaged by disease or trauma or aging. The use of our affinity therapy is an example of the use of biomaterials for therapeutic and even prophylactic applications previously reserved for pharmaceutical approaches. So we're happy to be working on this within DSM, and we see great potential for this application in the clinic in the years to come.